you, Bob. It's really quite an honor to be invited here for this purpose, not only because of the award in itself, but because of the really highly esteemed colleagues who participated in granting this great honor. I think Bob's introduction essentially covers my talk, so I think we can all go to the break now. But maybe I'll give you a few details instead. So we can just start by thinking about what one knows about the processing mechanisms in vision occurring at the very earliest stages of cortical visual processing. And over the talk, I want to develop for you a theme that beyond the fairly simple feed-forward properties that were described by Torsten Wiesel and David Hubel many years ago, we are now learning that the response properties of neurons and the function of neurons is actually much more complex, even at the very earliest stages of processing. Secondly, that this complexity is in part defined not only by intrinsic connections within a cortical area, which relate to this idea of the association field, but they are subject to top-down influences of attention, expectation, perceptual task, and efference copy. So neurons do not have a fixed functional role, but are constantly doing switching of their function on a moment-to-moment basis as we engage different tasks and different objects in our environment. And finally, we know that experience can shape these properties as a result of ongoing interaction with the world, and that we can see the effects of alterations in visual experience in these circuits that exist within the visual cortex and in the functional properties of neurons. And in particular, we look at an aspect of learning and memory known as perceptual learning, which we think actually involves the neurons in the primary visual cortex, among other areas. And so one has to think of the cerebral cortex as a whole as a learning engine capable of incorporating new strategies as we continue to experience the world. So to begin with, we know from the early work that neurons have receptive fields that are tiny windows on the world within which a stimulus will cause the neuron to fire. And depending on, in the primary visual cortex, depending on where the receptive field is relative to the center of gaze, you can have very tiny receptive fields near the center of gaze, and they get progressively larger as you go away. And so that defines the sensitive region of the neurons, but I will develop for you the idea that, in fact, neurons are sensitive to visual stimuli extending over much larger regions when you design the visual stimulus appropriately. But within the receptive field, it's known that a simple stimulus, such as a single oriented line segment, presented on the correct orientation will cause the neuron to fire. And then as you change the orientation, the response gets progressively less. So this then would imply that we take an image and break it up into a series of short oriented line segments that describes the contour boundary of that object. But that begs the question of how we assemble these elements into a unified percept of the global contour of the shape of this teddy bear. So one might have thought that this happens progressively over a hierarchy of cortical areas, going from the early visual cortex on into the temporal lobe, where you have neurons that are selective for objects as complex as faces. But what we've been learning is that even back at these earliest stages in the primary visual cortex, neurons are integrating much more complex aspects of the visual environment and are integrating aspects of contours that extend well outside the receptive field, which it sort of seems to be something of a contradiction in terms, because what is the receptive field except for the area of the sensory map where the neuron receives input? Well, the resolution comes in the difference between what we see when we use a simple stimulus to map this receptive field, such as a single oriented line segment. So here, if we put a line here, you'll get a certain response. You put a line outside this region, you get zero response. But when you use a more complex stimulus, such as putting two line segments into alignment with each other, then you find that you can get a response that is 2 or 3 times the response of the neuron from what you get with this simple line segment within the receptive field center. If you then do something to break the continuity between those two lines, you no longer get that sort of facilitation. So this, we think, relates to a general property of how we analyze the visual image, which was originally described by the Gestalt psychologists early in the 20th century. And what they noted, particularly the work of Max Wertheimer, was that there were certain rules by which we group stimuli in the visual scene, both the similarity, proximity, and a property that he referred to as good continuation. And that is that if we make a contour where there is relatively smoothness in the contour elements, so we tend to link this contour element with that one as belonging to the same contour, whereas this and this seem to belong to different contours. So that simplifies greatly the problem for how we parse the visual image. There may be a nearly infinite number of possible combinations of line segments within a complex image, such as what I'm looking at now, but we actually simplify it by only linking the elements that have this characteristic smoothness of contours. And this can also be expressed more geometrically by a concept that we call collinearity or co-circularity, and that is that object contours are relatively smooth. They don't take on sort of fractal characteristics. So if there is a straight line within an image, it tends to be kept straight for long distances. If there's a curved contour, it tends to maintain the same radius of curvature. And so at the level of the visual cortex, this describes the property of the association field. So you might have a cell whose receptive field is within this region, but it sets up a range of interactions across the cortical map where it links the contour elements that are collinear or co-circular with it. And this can also account for a property that, of contour saliency. So actually I should back up to point out this one, where if you take a relatively smooth contour and embed it in a complex background, it will pop out quite readily from that background. But if you have a contour where there's much more jumble in the relative orientation of the consecutive line elements and put that into such a background, you can't see it. It won't pop out. I mean, you can eventually find it through serial search, but it's much more difficult. And this is reflected in the properties of neurons in the visual cortex, as you can determine by having an animal trained to fixate a spot of light in the center of a screen, and you present these two patches of randomly oriented lines, and the task for the animal is to make a saccade towards the patch that contains this embedded salient contour. And we can look at how the responses of neurons in the superficial layers of the visual cortex are affected by the presence of this embedded contour. So we can either look at what happens when you just have a single line element within the receptive field of the receptive field's orientation, or this line of collinear contour segments as we put it either within the receptive field or in the opposite hemifield. And so what we find is that as we increase the number of lines that we put into alignment and putting into this sort of background, you get a progressively stronger response from the neurons. So there's a considerable contour-related facilitation. So we'll call this the contour-related response. And so so even in V1, in the primary visual cortex, the responsive neurons are as dependent on the global characteristics of contours that extend well outside the receptive field as they are on the attribute of the line segment that is presented in the receptive field center. Also, it's nice to point out that we know that there are inhibitory influences surrounding the receptive field, and so generally when we are looking at complex images, all of the stuff in the surround will suppress the response of the neuron. So that sort of begs the question of how neurons would respond at all if we're always looking at complex scenes. And this shows you how it happens, and that the global characteristics of the contour make it possible to lift the neuron out of this inhibition. So this is the response of a neuron to a line presented in isolation. This is what it responds to when the line is presented with the background, but then you get much less of this inhibition when you have this collinear contour. So we've been interested in thinking about the mechanisms of cortical processing in terms of the circuitry within the cortex. And this raises the finding that we had early on. So this is from the work that I did with Torsten Wiesel many years ago, where we discovered a class of connections within the visual cortex, and now this has been found to exist in every cortical region, whereby neurons can link activity across wide swaths of cortical territory. So if you look at this neuron, this is one where we recorded intracellularly from it and then injected it with a dye that spread throughout the dendritic and axonal arbor. So this is the dendrite of the neuron, and all of the rest of this is the axon of the neuron, which goes from the axon hillock at the bottom of the neuron and then branches many times, extending for long distances parallel to the cortical surface. And what this means is that since there is a visuotopic map on the cortical surface, what this means is that a cell over here is integrating information over an area of cortex that represents an area of visual space much larger than its so-called classical receptive field. So we think this can account for these rather higher order integrative properties of cortical neurons. One reason that to support this is in this curious clustering behavior of the axon collaterals. And so we can look at how this relates to the functional architecture of cortex by doing an experiment where we label a population of these horizontally projecting neurons and compare that to the pattern of orientation columns on the cortical surface. So what we do is we can use a viral vector to infect the neuron and transfer the gene encoding the green fluorescent protein into the neurons so that the neuron synthesizes tons of this fluorescent label, which spreads throughout its dendritic and axonal arbor. So this is from an experiment where we labeled about 6,000 neurons. And now you're looking down on the cortical surface through a confocal microscope, and you can see this pattern of projections where you can see these clustered axon collaterals. And when you superimpose that on the orientation map, so keep in mind that while neurons have the preference for orientation, there are groups of neurons extending from the cortical surface down to the white matter that share the same orientation preference. And as you go across the cortical surface, there's a regular clockwise or counterclockwise shift in orientation preference. So this is a false color image obtained with a technique known as intrinsic signal optical imaging, where the different colors represent the different orientations. So here we've made the injection in an orientation column representing horizontal, and the black lines are the reconstructions of the axon collaterals extending from the injection site. So within a short distance, these axon collaterals contact orientation columns of the full range of orientations. But as you get farther away, say about 500 microns away from the injection site, you see this clustering behavior that mediates connectivity between columns of similar orientation preference, as this is shown by this histogram. So this then adheres to one of the rules of good continuation, this sort of like-to-like connectivity between columns of similar orientation preference. And it also obeys the extent of these lateral interactions. So if you do psychophysical experiments looking at the distance over which these interactions can occur, if you make a contour where there's a certain, where you change the spacing between the contour elements, there's a certain critical distance beyond which you no longer see the salient contour. And the extent of that distance in visuotopic terms, when related back to the cortex, maps precisely to the extent of the horizontal connections in the visual cortex. So either if the spacing is too great or if the relative orientation is too great, then you no longer see these embedded contours. But when you allow these interactions to exist, they can propagate over multiple nodes in the network, and thereby allowing this phenomenon of saliency to extend over large distances of visual space. But we don't want to suggest that these interactions act constitutively, so that regardless of the behavioral context, they are always seen. But rather, they are subject to top-down influences of perceptual task. And I'll show you how this operates. But just to illustrate how this may be happening in your visual system, if you haven't seen this image before, it's probably difficult for you to know what is represented here. But if I allow you to have some measure of expectation operating, and I show you a more fully rendered image, and then the original image again, you can segment it quite readily. Now this demonstration will no longer work for any of you because you'll always have the expectation. So this is also seen in what we get from these contour-related responses in the visual cortex. So if we have the animal either do the contra detection task or an unrelated task in the opposite hemifield, we see different degrees of this contra-related facilitation. So about twice as much facilitation when he's doing the contra detection task as when he's doing the other task. And we think, so therefore these lateral interactions are subject to top-down influences of perceptual task. And we think that it is related specifically to the task of contra detection as opposed to attending to the location of the contour, because we can see what develops as we do these, as we train animals progressively on doing this test. So this is work, by the way, that was done by Wu Li, where he looked at the presence of the contour-related response. That is, how much facilitation you get as you increase the length of the contour when the animal is just trained to fixate. Just so he's not attending to the contour, he's fixating, and you see no contour-related response. We then train the animal to attend to the location of the contour, only he's not doing contour detection, he's just doing a brightness discrimination task. And there's no contour-related facilitation. We then train the animal to do the contour detection task, and you see a substantial degree of contour-related facilitation, which then is greatly reduced when he goes back to do the brightness discrimination task. And under anesthesia, there's no contour-related response. So a lot of these properties are important to study in awake behaving animals. And so the take-home message here is that this top-down signal carries information about the nature of the perceptual task being performed, not just attention, although Bob may want to put it under the rubric of attentional influences. I think it's important to think about the richness of information that is provided in this top-down fashion. And so we have the higher order integrative properties of the neurons, the fact that they are subject to top-down influences, and also now we want to point out that they are subject to learning. And this is something that we can see in the development of the response properties as the animal is trained on the task. Now, the phenomenon of learning has been divided into a number of subtypes, and this is from a diagram that was generated by Larry Squire, pointing out that memory includes both declarative memory, memories that we're conscious of, there's a memory of faces, facts, events, objects, and non-declarative memory or implicit memory, memories that we're not conscious of. And so this includes habit, skill learning. And in the perceptual domain, in the sensory domain, there's a form of learning known as perceptual learning. So it's the improvement in your ability to do a discrimination task by repeated practice of this task multiple times. So this is one particular task. So the contra detection task is one. This is another task, which we call a three-line bisection task, where we train subjects to determine whether the central of three parallel lines is located near the one on the left or the one on the right. And there's a certain offset from the central position in this task that is required for you to reliably see it as one way or the other. But this is a good task to illustrate some of the salient properties of perceptual learning, notably its specificity. So if I train you on this three-line bisection task, you'll be able to see much subtler differences in this offset of the central line after training over a couple of weeks. And so this is the threshold in the task, which has been reduced in the subject by about a factor of 2. But importantly, the training on this task doesn't transfer to a vernier discrimination task where the target line is in the same position in the visual field, same orientation, but where the context is changed to a collinear line as opposed to these two parallel lines. So there's no significant improvement here by training on the three-line bisection task. Yet you can then train on the vernier discrimination task and get considerable improvement. So whatever the underlying cortical mechanism is for this perceptual learning has to account for the specificity of perceptual learning. And specificity is something that had been originally described by Thorndike in the early 20th century. And because of that realization, it led to a change in the educational system in the United States, where it was thought that classical training, that is Latin, could be beneficial in learning anything, such as math. But what he pointed out, and which then subsequently caused this change in the educational system in the US was that if you wanted people to learn math, you train them on math, you don't train them on Latin. So here's this is a just as a stimulus array, which I will be referring to later. So we can train animals to do both the three-line bisection task and the veneer discrimination task. And we can see what happens when we present the same set of 5 lines, where in one case these 3 parallel lines are relevant for the task, and in another case the 3 collinear lines are relevant for the task. We'll get back to that in just a second, because I wanted to show you what happens with perceptual learning on the task. So as the animal, if you compare the performance of the animal on performing these tasks in the first couple of weeks of training versus the second couple of weeks of training, you see that there is a shift upward in what we call the psychometric curve. That is, as you increase the number of lines, if you look at this solid black line, the animal is able to do better at detecting the presence of the line with increasing length contours. But after a period of practice, you can do that job with fewer lines, and his performance improves. And along with this improvement in what we call the psychometric function, there's an improvement in the neurometric function. That is, this contour-related facilitation also shifts up from this level here to this level here. So it seems that even in the primary visual cortex, the response properties of neurons are not fixed, but they are capable of undergoing change as a result of learning and experience. I just wanted to point out, well, actually, maybe I'll just skip over this because I want to move on to another example that shows what happens in the responses of neurons when you're not doing a contour detection task, but when you're doing a shape discrimination task. So here is where we cue the animal to look for a particular shape, and this is work that was done by Justin McManus. The shapes include a circle, or a wave-like shape, or a straight line. And the question we asked is how that influences the shape selectivity of neurons in the visual cortex. So after presenting the contour, at the end of the trial, we have this contour embedded in one of these complex backgrounds, and the animal is supposed to make his saccade, his eye movement, towards the embedded contour, towards the contour that is cued to detect. But before we show the target stimulus, we have a series of presentations of a set of stimuli where we have a line presented within the receptive field and then move other line elements outside the receptive field to different positions and orientations to find out what is the optimum shape that the neuron that we're recording from is responding to. And then we represent that shape selectivity on a heat map where different positions in the map correspond to different shapes. So the result of this experiment is illustrated as follows. So here is where we have a neuron that we've recorded from while the animal is looking for the straight line shape. And what we find is that the optimum stimulus, and these heat maps show the nature of the stimulus that is at any particular location by this cartoon of line segments. So this shows that this neuron responds most to a contour made out of a series of nearly collinear line segments. But then when you cue the animal to look for a circle, the optimum shape changes to one involving more of a co-circular sequence of line segments. And when he's looking for the wave-like shape, you see even a more elaborate shape selectivity of the neuron, where you see this reversal in the radius of curvature, so the neuron becomes selective for more of a wave-like shape. And we see the entire population of recorded neurons shifting from one position in shape space to another, depending on the shape that the animal is looking for. So once again, it shows that the neuron's response characteristics are selected for more complex geometries than just short-oriented line segments or even just one extended straight line, but it depends on the expectation of the animal. So although we're dealing with fairly similar simple stimuli here, it's important to point out that the functional properties of neurons are constantly shifting, changing according to the expectation of the animal.
Question. What's the time course of these changes? How quickly can
we find that it can change on a trial-by-trial basis, but it's sort of is a good question to ask, how rapidly, if you could sort of do a task switch in the middle of stimulus presentation, how quickly could you see it? But at least, which we don't know, but at least it seems if we do two parallel maps of the receptive field where on alternate trials we shift the expectation of the animal, we see that the properties converge to different shapes on the two parallel streams. So yeah, so it's important to point out that, I talked about perceptual learning altering the response properties of neurons. So this is within a long-term effect, what we could put under the rubric of plasticity. But the reason that I use the term the dynamic brain, and we talk about dynamics of circuits, is this is something that doesn't necessarily require changing synaptic weights or making new axonal connections, but it's something that occurs quite rapidly within the context of an existing anatomical framework. And so we call this more dynamic properties. And as a consequence, we think of a neuron, rather than having a fixed function, as being an adaptive processor capable of changing its functions in accordance with the needs of the current perceptual task, and where it gates its inputs, where it can sort of suppress inputs that are relevant for the task being performed and select components of its association field that are task relevant, and thereby taking on these different selectivities. So this slide is to illustrate what we think may be happening at the circuit level, where one should think broadly not just of the intrinsic connections within a cortical area, but how cortical areas interact with one another. So the classical view of cortical processing is one of a feed-forward network of connections going from the primary visual cortex through this ventral pathway for object recognition, going from V1 to V2 to V4 and into the infratemporal cortex, and a dorsal pathway, the so-called vision, using vision for guiding movements, and that goes into the parietal and ultimately into the frontal lobes. So if we just consider this pathway, or in fact any other pathway, what is notable is that for every connection where there's a feed-forward connection, there is a feedback connection. So higher order cortical areas are communicating some information to the antecedent areas. And we think it's important to consider the possibility that the information that is being communicated in this feedback fashion are these top-down influences carrying information about perceptual task, expectation, and attention. Although we are agnostic as to the possibility of participation of other pathways. So in addition to these direct connections between cortical areas, and I also include this one for Kathy Rockland, who showed that there's also, you can also hop across the entire pathway and go directly from the infratemporal cortex to V1. But the strongest feedback to V1 is from V2. So one can then think that that this set of interactions across cortical areas includes the map of the horizontal circuit, so to say, that describes the association field within each cortical area, and that there are these interactions, these brown lines showing the feedback connections between the different cortical areas, that allows the neurons to gate their inputs across the whole swath of cortical areas, and that overall the whole set of cortical areas are changing their state according to the perceptual task that is being performed, and the actual functional role of these areas is changing in this fashion. So we were just interested in considering how this process of gating of connections might be possible. The ideal situation would be if you could have an axon from one cortical area contact the axon terminal in the antecedent cortical area and turn it on and turn it off. But that is not an anatomical reality. And so we have to think of how this could happen more at the network level. So Valentin Pike attempted to create a biologically realistic model with neurons with the normal biophysical properties, where we have this feedback gain control that can influence how the neurons interact with each other. And we extended the network to include many groups of orientation columns. It's a full set of orientation columns is what is known as a hypercolumn. So here, each of these nodes in the network, and these are heavily interconnected via the horizontal connections, describing the association field, which are excitatory connections between columns, as well as inhibitory connections in the opposite direction. So if you then expand this network up to include maybe 500 hypercolumns, you can see how it operates in capturing the contours present in natural scenes. And so in this image, which is a often used image for computer vision, the image of Lena, what we find is that when we have this network operating on the image where we can include the long-range horizontal connections and a strong feedback gain, the network captures all the contours present in the image and sort of maintains a good dynamic range between contours of different contrast or different strength. And it also replicates what we see experimentally in the response properties of neurons. First of all, so here is this top-down input that is reflected in this bright orange area, which then facilitates the responses of the neurons whose receptive fields fall along the contour and inhibits the response of neurons in the flanking regions. And this is what we see with, when we do large-scale recordings. And it also, interestingly, replicates we see in terms of the timing of these responses. So one thing I didn't point out to you is that this contour-related response occurs with a certain delay after the initial visual response. And this has been attributed variously to the time required for the information to be fed back from higher order areas or for information to be propagated over a fairly slow horizontal connections. But what this model shows is that it's due to neither. It's actually due to the time required for these foreground-background interactions to take place, for the neuron to go from one stable state, for the network to go from one stable state to another. And so this shows this then in the model, which then replicates the delay as well as the contour-related response for both excitatory and inhibitory neurons. So the question is, can we get any evidence in support of this idea that the connectivity within the network can be dynamically expressed? So to do this, an experiment that was done by Nirmala Ramalingam was to embed, we embedded these arrays of electrodes in the visual cortex chronically, so that on a day-by-day basis we could collect the information from hundreds of neurons simultaneously. And importantly, we were able to use a statistical technique to look at the interactions between cortical sites. And because we could take advantage of the functional architecture of the cortex, that is the visuotopic map of the visual cortex, we could distinguish between cortical sites representing contour elements that were relevant for the task being performed versus those that were task irrelevant. So here is where we have this 5-line stimulus that the animal can either be cued to do the 3-line bisection task or the vernier discrimination task. So when he's doing the 3-line bisection task, these lines are task relevant, these lines are task irrelevant. And if this now shows you the recording sites from the electrode array, each of these points represents each of the receptive field position of at each electrode. And these gray bars represent the stimuli that we use. So we have 3 different positions of the parallel lines, or 5 different positions of the collinear lines. Actually, there's 5 different positions of the parallel lines. So there's a total of 25 different stimulus conditions. But on top of that, the animal can be doing either the 3-line bisection task or the vernier discrimination task with the identical visual stimulus. He's just cued before the stimulus is presented as to which task he's going to perform. And we can look at the function of the neuron, which is defined in terms of the tuning characteristics of the neuron for various attributes in the stimulus. So here we're looking at how a neuron responds to a changing offset of the parallel lines that are placed outside the receptive field. When the animal is doing the 3-line bisection task, where those 3 parallel lines are task relevant, we see a substantial degree of modulation in the response of neurons. But when he's doing the vernier discrimination task, when those 3 lines of task are irrelevant, there is much less modulation in the response of neurons. And this, by the way, this difference between these 2 points occurs from the very outset of the response of the neuron. So here, where we don't have this complex background, the contextual influence arises from the very first output of primary visual cortex. And so one can do this one can see this both with recordings based on spiking activity of individually recorded neurons, or based on local field potentials, which reflect the combined activity of hundreds or thousands of neurons at the same time. And there too, we see this differential tuning depending on which task the animal is performing. And over the population, this can be expressed in terms of mutual information. That is, there's more information conveyed by the response of the neuron relative to the attributes in the task that are relevant for that task when he's doing that task compared to when he's doing a different task. So there's a shift in the amount of in the kind of information that is conveyed by the neuron. And so one could imagine that, well, maybe one way this is done would be to just turn off the activity of the neurons that are irrelevant to the task. But that's not what happens. These are the responses that one measures at the flanking sites, and there's no difference in the responses depending on the task the animal's performing. But rather, there is a change in the way in which these sites interact. And we can measure this by one of 2 techniques, either cross-correlation analysis, where we look at the relative timing of spikes between pairs of neurons between the 2 different cortical sites. And when the 2 are interacting, when there's a neural interaction, there's a peak in the correlogram, such as shown here. And when they're independent, when there's no neural interaction, the correlogram is flat. And so what we find is that between the 2 sites, with the same amount of spikes occurring between the neurons, you'll either get an interaction occurring, a peak in the correlogram, or a flat correlogram, dependent on the perceptual task that the animal's performing. Remember, the stimulus is identical. So the only thing that is changing is what the animal is thinking about, what task he's performing. And you can see this change in interaction also with another measure, Lfp Coherence. So we look at how the local field potentials either covary or vary independently. And interestingly, if we record from 2 sites that are representing the central line and the flanking line in the 3-line bisection task, you see much stronger Lfp coherence when he's doing the 3-line bisection task than when he's doing the irrelevant task, the vernier discrimination task. But curiously, we see the opposite relationship when he's doing we're recording from the collinear sites when he's doing the vernier discrimination task compared with the 3-line bisection test. So there's much stronger coherence when they are task irrelevant. So what could account for that? We think it has to do with the perceptual strategy and going back to the work of the Gestalt psychologist. And that is that the 3-line bisection task is more akin to a grouping task based on proximity. And the vernier discrimination task is actually a segmentation task where you're trying to separate the collinear line segments from one another to be able to see this offset between them. So what happens if we look at the coherence between collinear sites in a task that involves perceptual grouping, namely the Contra detection task? And what we see is that, in fact, so here is the experimental design where the animal is fixating here, and he's either going to do the Contra detection task, as shown here, or the 3-line bisection task, where he's not paying attention to the embedded contour. And what we see is that there is stronger coherence between collinear sites when he's doing the Contra detection task than when he's doing the other task or when he's just fixating and not doing either task. And another interesting thing to point out is that these changes in coherence are not dependent on the stimulus, but dependent on the task. So we see this difference in coherence before the stimulus is turned on. So the stimulus is turned on here, but the animal has already been cued earlier as to which task he's going to perform, and you can see these differences in coherence happening and sort of setting the cortical state to be ready to analyze the visual stimulus in a way that is appropriate for the stimulus for the task that he's performing. Also, there's some interesting features about how the population activity carries information about the stimulus above and beyond what you see in these changes in tuning. But actually, unfortunately, Nancy's not here today, but I just wanted to bring this slide up, which shows that brings up the idea that because the properties of neurons are not fixed and they can change their functional role within a broad range of functions, this sort of raises the possibility that when you're doing fMRI, it can explain for why you see a lot of seemingly unrelated functions in the same cortical region. So now, to end with, I wanted to tell you to shift from the idea of short-term dynamics to long-term plasticity. So here we've sort of painted a picture that there's this existing anatomical framework that neurons can multiplex their function or change their function depending on this top-down signal, which we think involves an interaction between cortical feedback and intrinsic cortical circuits. But we also know that experience can induce long-term changes in cortical function. We saw it in the example of perceptual learning, but we've also been working with another model of experience-dependent plasticity. But now keep in mind that this is in the adult animal. So from the work that Torsten and David did many years ago, they found that there was a critical period in postnatal life during which the neurons could change their functional properties and circuits, namely thalamocortical circuits, could change as a result of altering the balance of input between the two eyes. But this now shows some experiments that illustrate another set of properties and another class of cortical connections that retain the capacity for plasticity throughout life. And what we've done here is to do a rather extreme version of altering visual experience, which is to make a focal retinal lesion binocularly in corresponding positions in the two eyes. So what this does is this removes visual input from a well-circumscribed region of the visual cortex. And so the expectation would be that after you make those lesions, that area of cortex is silenced for the rest of the life of the animal. But what we found, quite to the contrary, is that neurons whose receptive fields were originally within the part of the retina that later became part of the retinal scotoma, the blind spot that we induced with a laser, they shift their receptive fields to intact regions of the retina. And so this is the receptive field positions that we see when we implant electrodes, use these chronically implanted electrode arrays. And by the way, the advantage of this approach to looking at the phenomenon is that you no longer are subject to selecting the few active neurons that are still there after all the rest of them have been silenced. The electrode is fixed. We haven't moved it around to find an active neuron. We find that 90% of the cortical sites recover visually driven activity within a period of weeks following the lesion. So what this means is that this represents the lesion part of the retina and the area of cortex that received input from the lesion part of the retina, so that initially that area of cortex is silenced, but over a period of time there's a shrinkage in the representation of the lesion part of the retina and an expansion in the representation of the surrounding part of the retina. And we think this may have a useful function for visual recovery after lesions of the retina. And this is a model that shows what one might expect to happen in a case of macular degeneration. So this is the most common form of blindness in the adult. And if you were to sort of present an image that you had to look through one of a corrupted retina, such that you have with macular degeneration, where there are regions of geographic atrophy and in between the salt and pepper loss of photoreceptors, you would have difficulty in figuring out what the image was if you had to look at it through that retina. But if you allow the shifting receptive fields where they maintain their original line label for their original position in visual space, that then allows a process of perceptual fill-in, so though locally the image may be somewhat corrupted, globally you can make out the image quite well. And so we think this plasticity, which may use the pre-existing association field, which operates for contour integration and shape discrimination throughout life, can be re-invoked for functional recovery after lesions of the central nervous system, such as what may occur with stroke or neurodegenerative disease. And to look at what the circuit changes are that underlie this phenomenon, we used a technique of two-photon microscopy, which we developed for large animal imaging with Winfried Denk. And what the technique involves, if you're probably, I'm talking to the cognoscenti here, but it is a technique that allows you to see with high resolution fine structural details of neurons in below the cortical surface down to a depth of up to 500 microns or so. So we can image neurons throughout the superficial cortical layers. We label them with a fluorescent tag, so by infecting them with adeno-associated virus containing the green fluorescent protein gene. And after allowing enough time for that gene to be expressed, we can then see these neurons in their great detail, and I'll show you an image in a second, both the full dendritic and axonal arbors of these neurons. And in the context of the retinal lesion experiment, what we do is make several injections of the virus along a line of injection sites, and then we can image the pattern of projections from the injection site into one side or another of the line. And then after getting a baseline map of the circuit in the visual cortex, we make a retinal lesion where the lesion boundary is placed right to one side of that set of injections, so that the projections to this side correspond to the lesion projection zone, and the projections to this side correspond to projections in normal visual cortex. So first of all, this shows you what the two-photon images look like. So this is in the living brain. This is in the injection site. You can make out the spines from the dendrites of neurons, and you can see these clusters of axon collaterals. And notice this sort of blebbing along the course of the axon. So these are known as axonal boutons, and they're the site of presynaptic. These are the presynaptic sites along the axon. Now, when we don't do any lesion, just to look from week to week at these axonal boutons, we see that they are constantly disappearing and appearing at a rate of about 7% per week. So this is this rather extraordinary phenomenon that the circuits within our brains are not fixed, but the synapses are constantly undergoing this churning throughout life. And we don't know whether there may be some synapses that are more relatively stable, but at least over the period of time that we imaged, we saw this turnover occurring at that rate. You'd see, over 3 weeks, 21% new boutons and 21% disappearing. But without making a lesion, the branching pattern seems to be relatively stable. So this is what we see when we make a retinal lesion and look in the lesion projection zone. So again, we've made an injection to one side, and we're looking at the lesion projection zone at different time points before, on the day of, and after we make the lesion. And what was quite extraordinary is that on the same day that we make the lesion, we see this extraordinary outgrowth of new axons. So these yellow traced axons are the ones that have grown relative to the previous imaging point, and the red ones are the ones that disappear relative to the previous imaging point. And you can see that on a week-by-week basis, there is constant new outgrowth and pruning occurring in parallel, with a net increase in the density of these horizontal connections in the lesion projection zone over the imaging period. And we think this then corresponds very closely to what we see in terms, like when we record with these arrays, to the rather rapid initial change in the receptive fields of neurons that we see on the same day as we make the lesion, and the eventual remapping of the visual cortex that occurs invading the central of the towards the center of the lesion projection zone. And we can compare what happens to these excitatory connections to what happens with inhibitory connections by using a virus that Sally Marek had constructed that is under the control of the GAD65 promoter. So we can then selectively image inhibitory neurons. And this just shows that by co-labeling neurons with the GAD65 AAV virus and a cocktail of calcium binding proteins, which labels 99% of inhibitory neurons, that we see co-localization, and therefore we think that we are selectively labeling inhibitory neurons. And this shows that you get a similar sort of efflorescence and pruning of axons of inhibitory neurons within the lesion projection zone. We have made a similar series of experiments in a mouse model that allows us to more take a detailed look at the molecular mechanisms underlying this plasticity. And so this one is one involving the whisker barrel system of the mouse. So the positions of the whiskers are mapped in the somatosensory cortex of the mouse in terms of a series of rows of barrels. And one can induce plasticity, and we can create a lesion projection zone by plucking the whiskers. So we pluck the D and E rows of whiskers. And what happens is that over a period of time, these barrels, which are initially silenced, then become responsive to input from the C row of whiskers. So there's, again, a remapping that is taking place. And we can see that this remapping is associated with a sprouting of excitatory connections into the deprived barrels and a sprouting of inhibitory neurons whose axons are within who are resident within the deprived barrels towards the source of this new excitatory input, which we think is useful for maintaining a balance between excitation. So here's the balance of excitation and inhibition. So here's the sprouting of excitatory input into the barrels, and here's the sprouting of inhibitory connections from the barrels. And we can then see what the molecular mechanism may be, and this is something we've seen both in the monkey visual cortex following retinal lesions and in the mouse somatosensory cortex following whisker plucking, that these orange labeled neurons are ones expressing BDNF and the brain-derived neurotrophic factor. And so these are localized to the deprived barrels, and we think therefore is the stimulus for the sprouting of connections into the lesion projection zone. But also keep in mind that we see this phenomenon of pruning of connections. And this is, and for this we've looked from after being inspired by some of the work of Mark Tessier-Levine, we've looked at the apoptotic pathway. So a signaling pathway that is involved in cell death early in development does not lead to cell death in the adult, but as I'll show you, is responsible for pruning of connections. So, and this is where it's useful to have a mouse model from time to time until Bob comes along with the monkey knockouts that we need. If we knock out, so there's an interaction between the neurotrophin signaling pathway and a receptor known as death receptor 6, this guy here. And that leads to the activation of caspases, which then leads to axonal retraction. So if we knock out death receptor 6, so this is what you see normally. There's this sprouting of new connections in yellow, pruning of old connections in red that occurs progressively from day to day in the mouse somatosensory cortex. But that pruning entirely disappears in the Bax knockout. And so it appears that this pathway may be useful for the sculpting of connections. And interestingly, we see what seems to be even a heightened outgrowth of connections when we make these knockouts along this pathway. So, to summarize, there seems to be different levels of dynamics or plasticity that occurs within the visual cortex, and this is seen not just in higher order cortical areas that are thought to represent declarative memory, but is a dynamic process that can even affect neurons at the very earliest stages of visual cortical processing. And one may be concerned that if the primary sensory area is so dynamic, why doesn't that disrupt your ability to see things in a stable visual environment? And the answer to that is because of the fact that the alterations in function are only seen when you're doing the trained task. So these properties are just available in a just-in-time fashion that when you're doing a Contra detection task or a 3-line bisection task, the learned properties are manifest only when you're doing the task. So that this top-down interaction is required for both the encoding of the learned information and for its recall. And since I'm in this context of a general lecture, I like to speculate more broadly that this may raise a general theory of brain interactions, of the circuit mechanisms of brain function. That every cortical area has represented within it an association field. And by these top-down interactions, neurons can select components of their association fields and take on different functional roles in ways that are appropriate for the task at hand, suppressing task-irrelevant information and gating, allowing the input from task-relevant information. And this idea of interactions between global cortical circuits and local circuits, this idea that feedback gates the local circuits, is something that may operate not just in cortical interactions, but in interactions between the hippocampus and the cortex, where the hippocampus can be thought of as being a pointer for linking bits of information across the cortical maps, where there it can be used for more rapid learning, but also its role can be decoupled after consolidation. And for interactions between the striatum and say the motor cortex or prefrontal cortex, where again the striatum would serve a pointer function for sequencing operations that occur with habit learning. And so we think this may be a sort of a useful theoretical framework to think about how circuits can contribute to cortical function and for the dynamics of cortical function. And so I just wanted to acknowledge a number of my collaborators. This is a sort of a historically long list, but in particular, Wu Li had pioneered in doing some of the most wonderful experiments where we look at the properties of neurons in the monkey in different behavioral contexts. Valentin Pike had done this very nice computational model, which we hope to extend in the future to see how we can imagine how the dynamics in input selection can occur for more specific tasks. And of course, Torsten was involved early on in identifying this important class of connections, which we think are playing a critical role in mediating the association field and its gating. And we have enjoyed the input from Gerald Westheimer in doing the psychophysical experiments, from Winfried Denk in designing and building a new 2-photon imaging system, and Shimon Ullman for some of the modeling work that we've done. So it's sort of it takes a community to actually successfully pursue these experiments that have to be done in a highly collaborative and interdisciplinary way. So I thank you all for coming, and open for questions.